Good afternoon. Welcome back. We're delighted to have you all here today. A wonderful set of presentations this morning. I think they'll be exceeded by those this afternoon. This is just the gift that keeps on giving. We'll have only 3 talks this afternoon. Unfortunately, as Ralph pointed out this morning, Paul Weiss is not able to come. But I'm Nick Spitzer, the director of the Kavli Institute for Brain and Mind, which oversees the Center for Brain Activity Mapping, and Ralph and I have enjoyed working on putting this together. I'm very pleased to introduce a local friend who didn't have to get on an airplane to come these many miles, Erkki Ruoslahti from the Sanford Burnham Medical Research Institute, and his talk title is in front of us, Targeting of the Brain and Its Diseases with Nanoparticles. Erkki.
Thank you. So my lab has lately worked on targeting of the vasculature in various diseases. And we like to use nanoparticles because peptides, which is what we use for the targeting, they tend to have only moderate affinities, and the avidity that the nanoparticle presentation provides is useful in getting good targeting. So we've mostly done work on cancer over the years, some on atherosclerosis and a little bit on inflammation. But we've now gotten into brain research, and I'm a little uncertain about my fit to this program because I've only been a neurobiologist now for a year. But we have some new targeting results. The way this came about was that the DARPA, one of their offices, advertised a grant program that Mike Saylor, Sangeeta Bhatia, and I put in an application for. We have always had a joint grant since Sangeeta was here at UCSD, and at the time this program became announced, we didn't have a joint grant. And now we do. So in this project, we target brain injuries, and I'll talk a little bit about that in a moment. Now, as we interacted with the DARPA people, I talked to them about the possibility of targeting Alzheimer's, and they got excited about it and asked me to write a proposal, which I did. So now I have 2 of these DARPA contracts or grants, and this one I collaborate in with Stuart Lipton, who is a card-carrying neurobiologist and neurologist who actually practices here at UCSD. And most of the work I'm going to talk about was done by Aman Mann and Pablo Skodeler, and Shikan Shi and Gary Brown provided some help to them. So the way we target the vasculature, and as you'll see in a moment, the parenchymal tissue in the brain as well in these diseases, is that we use phage display in vivo. So we nowadays use T7 phage, where the peptide that we express is on the coat protein of the phage, and there are 415 coat protein molecules, and we modify about half of them. One phage particle carries one peptide. Then the issue becomes, how are you going to find those phage particles that carry a peptide you're interested in? And the way we do this is we inject the library, which has about 1 billion peptides, IV into a live mouse. And the idea is that this way we probe the vasculature and whatever else can pick up, bind the peptides in its natural state inside the body. We collect the tissue of interest, and in this case the brain, and then sequence the inserts nowadays using high throughput sequencing. This is a cartoon showing what happens if the phage particle carries a peptide that has the ability to bind to the vessels or penetrate into the tissue that is of interest to us, then those particular particles will concentrate at that target, and that will show up in the sequencing. We actually, when we first developed this technique almost 20 years ago, we had the brain and the kidney as our targets. The purpose of this paper was just to show that this could be done. And it actually worked really nicely. We have a few brain-homing peptides that we haven't really done anything further with. Other people have since used the same method to find additional peptides that target the vasculature of the normal brain. So our task then was to target brain injuries. And we set up models. This one was set up with the help of Stan Krajewski at the Sanford Burnham. It consists of 9 needle punctures in the brain on one hemisphere, and they go in about 3 millimeters, close to the corpus callosum, which is affected as shown here in the injury side and pretty much normal on the uninjured hemisphere. So we screen in this system, and much to our surprise, got a result that we have never had before. And that is that we had, just in one round of homing, an overwhelming number of a peptide that we have given the code name B.R.A.I.N for brain injury emergency peptide. And it was about 40%, this peptide and its variations were about 40% of all the phage in that screen, which is just a remarkable result. Usually we get much lower numbers of specific homing peptides. Then when we make the synthetic peptide and label it with fluorescein, inject it IV, it nicely localizes in the injury area, as shown here. Here's a control peptide, nothing there. If there's no injury, there's no homing. And of course, this is just the absolute negative control. Here we just look at the brain on a light table under fluorescent light. So there's a lot of fluorescence there in the brain. In a section or slice of the brain here, here's the injury. Again, here's a control peptide. B.R.A.I.N has a free cysteine, and it's also a linear peptide, so we've used this peptide mostly as the control. And later on, when you see a control mentioned in my slides, it's this peptide. We can get the homing into the injury itself at 6 hours after injury, but for some reasons that we don't fully understand, at 24 hours after the injury, there is more fluorescence elsewhere. It's still specific, but it's not really in the injury area any longer. And so something happens in the injury area that seems to prevent the peptide from getting there, but the rest of the brain is affected, and it goes there. This type of results have actually given us hope that we might be able to target concussion also, because we do get some accumulation of this peptide in the healthy or uninjured. In this case, it's not really healthy hemisphere. And that hemisphere, of course, has mostly just gotten injured indirectly. So we're setting up a concussion model to study this. We can also do non-penetrating traumatic brain injury, and this peptide homes there as well. I didn't put in slides on that. So we then wanted to look at this peptide a little bit more closely in the brain, and we set up an overlay assay where we use silver particles. They were designed by Gary Brown in the lab, and this paper will actually come out next week. They're very nice in a number of ways. One which we're making use of here is that when you couple a fluorophore on the surface of these particles, the emission from the fluorophore is greatly enhanced. And these particles are very bright. Gary has also developed a way of etching them when they're outside a cell and such, but that's not really relevant here. What is relevant is that when we just put the nanoparticles on a glass slide and look at it under the microscope, we can actually see the individual particles, or what we think, based on a number of reasons, are the individual particles. So we use these nanoparticles then in overlay assays to see if we could get these particles to bind to some structures in the brain. And indeed we do. And at this magnification, it's hard to see the particles. And I have a higher magnification. I just put this in to show that the controls are negative. So here's a better view of what the injury area looks like. And this is the corpus callosum. That's where the probe mostly concentrates, even on in these overlays. Now, we then, of course, looked at the normal brain as well. And the target for these particles clearly is present in the normal brain. It's, these are pretty much representative images, so clearly it's upregulated or it's somehow more available in the injury brain sections than normal, but it is in the normal. So one conclusion from this is that we seem to be targeting something that is present in the normal brain, but upregulated in the injury area, and the peptide has access to it during a period during a short period after the injury, because the blood-brain barrier is actually lost at that time. So everything penetrates there. What this peptide clearly does is it's more effective in accumulating there, and it stays there longer than things that just penetrate there non-specifically. Now, this observation also pleased us in one way, and that is that we want to identify the target for this peptide. And it would have been logistically almost impossible to get enough of this injury brain material to isolate anything from it for receptor isolation. But now we can use normal brains, and that work is underway now. Now, we also wanted to look at this, the homing of this peptide using this new technique, clarity, where the brain is treated in a certain way. It takes a month of treatments, but it becomes essentially transparent, as you see here. This was published just a year ago, and these people actually have absolutely fantastic images of brains done with this technique. It's set up here at UCSD, and that's where we have been doing this work. We've been somewhat restricted by computer power because this is extremely power intense in terms of computer power. But here's first to orient you, a clarity cleared brain stained for nuclei, and here's the injury. And here's the repeptide homing to the injury area. And here's a control where we don't see much in this particular section. Now we then, in some other experiments, we do see some homing of the control peptide, or it isn't really homing, it's just that everything penetrates into the injury areas. But it's vastly more, there's vastly more fluorescence in the specific homing peptide. And here's the, we wanted to look at the corpus callosum and took several optical sections from that area. And just to orient you again, here's the corpus callosum, here's some other structures in the injury was again up here. And here's the just a fluorescent image from the clarity cleared brain. And now I hope I'm going to be able to show you how these images can then be. And here's just what you might consider a thick slice of the area, not the whole brain, but you can really see how the fibers go there and how the peptide attaches to them. So that's where we are with this B.R.A.I.N peptide. We're isolating the receptor and judging from our ability earlier on to isolate peptides, receptors for peptides that home to tumors, I think we can accomplish it. And we want to then see whether that target protein or whatever the target is more exposed in the injured brain or upregulated, and what its role might be in the inflammatory process or injury process. So the other project targeting Alzheimer's, then as I mentioned, Stuart Lipton is our collaborator. We use mostly a mouse model that is called J20. These mice have 2 mutations in the amyloid precursor protein, and they develop a disease really early on. They have some symptoms or signs or both already at 3 months, such as neuronal loss and changes in long-term potentiation. The additional lesions and impairment then appear later, and at 9 months of age, they pretty much have a full-blown disease. This is in contrast with some other models where there's only 1 mutation in the protein, and it takes longer to develop the disease. So I sold this project to the DARPA people as a vascular targeting project, because that's usually what the phage targets. The phage is a nanoparticle, and if the vasculature is intact, it's not really going to get out of the vasculature. And that was our original hope, because we wanted to target the vasculature. But it turns out that there's quite a bit of literature to the effect that the vasculature in Alzheimer's lesions is compromised, and that the blood-brain barrier is part of that compromised function. We actually ended up finding that when we did a screening with the Alzheimer's mice and compared them with wild-type mice, counting the phage that we recovered from the brain, we recovered 5 times, sometimes in some experiments even more than 5 times the amount of phage we recovered from wild-type mice. And our explanation is that we're not just targeting the vasculature, but the phage is getting out of the vasculature and also probing the parenchymal brain tissue. We only collect here the hippocampus, which is the area of the brain mostly affected in Alzheimer's disease. So we found 2 peptides from this screen that showed selectivity for Alzheimer's compared to normal. Very good selectivity, as you see here. I'm showing one that we codenamed CES. The other one, which we call Casper, gives pretty much the same result. So I didn't put in a slide on that. When we then make the peptide and inject it IV, and that phage experiment was also from an IV injection. When we inject the synthetic peptides labeled with fluorescein to mice, in the Alzheimer's mice, we find that, and it's not very clear here, but there's fluorescence in the dentate gyrus, in this case, of the hippocampus, and it's clearer with Casper, and the controls are negative. So here we then did more experiments with Casper because it seemed to be a stronger homing peptide than CES. And we again resorted to that overlay to learn more about the peptide and its target. And here, instead of using fluorescent silver nanoparticles, we now detect them with silver staining. And what you see here is that Casper, the Casper nanoparticles do stain the dentate gyrus in the Alzheimer tissue, whereas the control nanoparticles do not, and Casper does not stain the wild-type tissue, and nor does the control, of course. So here we seem to have a different situation than with the B.R.A.I.N peptide. This, whatever the target is for this peptide, appears to be specifically upregulated in Alzheimer's. We also, that was the dentate gyrus. Here we looked at an area called alveus or alveolus, which is also part of the hippocampus, and found actually these really intriguing structures, long rows of nanoparticles, which seem to be binding to a structure that is also in the control peptide sections, although the control nanoparticles do not bind to it. And I haven't been in this business long enough to know what these structures might be, and Stuart hasn't really seen all this data. He's been traveling so much lately. So maybe some of you can tell me what they might be. But they look intriguing, and they're very clear in a higher magnification here. Maybe some sort of a nerve fiber bundle. I don't think they are blood vessels, but they could be that too. So that's where we are with those peptides. And again, we're tooling up to isolate the receptors for the peptides, which we think, based on our results, are specific for, specifically expressed in Alzheimer's brain. Now, we then did another screen where we compared the mice at 3, 5, 7, and 9 months as you may remember, the disease first manifests itself already at 3 months of age, and is fully developed at 9 months. So we found a very common sequence motif among all these 4 samples that interestingly was most common in the 3 and 5 month samples, indicating that it may be this sequence motif may be binding to an early manifestation of the disease, which of course would be very useful if it holds up. Now, this was just a sequence motif, so we looked at the 20-some peptides that had the motif, and then made, and I forgot to point out what is written here, that there was very little of this sequence motif in the phage that we recovered from normal brain. So that was promising. So we made a peptide that incorporates the most common features of this motif, and we call it the DAG peptide. We originally called it Dagger, but the DARPA people, one of them in particular, didn't like it, so we had to go to DAG. I like Dagger better. Then again, overlay here is very impressive with the DAG peptide. Here's the Alzheimer dentate gyrus, and here's wild-type dentate gyrus. Big difference, somewhere around 13-fold in this case, if I remember correctly. We also found that the cell layer called CA1 is positive for DAG, and again, there is little binding of the nanoparticles to the control. Now, some of this may be background, but nonetheless, the difference is, again, somewhere there between 10 and 15-fold, which I think would be quite adequate for specific targeting. And we know from tumor studies that that is the case. So this synthetic peptide then also specifically homes to these structures from an IV injection, as shown here. There's one particularly positive structure, maybe one cell, and well, I won't get into that. The next slide shows it better. What is shown here is that the controls, again, are negative. Now, here's a better view of the comparison of the blood vessel molecule CD31, FAM-DAG, and then the nuclei have been stained blue. So here's again the dentate gyrus, and you can see that there's a lot of accumulation of the peptide there, and a lot of these structures are yellow, which indicates that there's a co-localization of FAM-DAG with blood vessels. When we look at this more closely, we see structures like this. Here's something that has to do with the blood vessel. It's red. And here's just one cell hugging the blood vessel. So we think that this is an astrocyte, and this is actually what is called the neurovascular unit. It's another view of a similar situation, not quite as close an association between the fluorescence positive cell and the blood vessel structure. But they again seem to be going together. So it looks like what we have here is a target molecule that is expressed specifically in the dentate gyrus and the CA structure of the hippocampus in Alzheimer's, but not in normal mice. And that the peptide detects something in the neuronal cells or in association with them, but it also detects the neurovascular unit. And I'm particularly excited about this because, well, first I focus a lot on the vasculature, but this indicates that there's a specific change in the neurovascular unit, and that we may be able to detect it very early on by using this peptide for, say, imaging. And again, we of course need to identify the target. So what happened in this screen was that we initially thought we'd be targeting something that is specific for the blood vessels in Alzheimer's. And we sort of found that because the neurovascular unit is at least close to the vasculature, or a part of the vasculature in a way. But we also ended up probing the parenchyma in the diseased brain. So when we started this work, a big concern was how we were going to get through the blood-brain barrier to be able to target the neurons, which the DARPA people felt were the important target. Now we know that we don't have to do anything to the blood-brain barrier. It's not there, and we can probe the parenchymal tissue. And we're going to do more screens to find more targets, but that's where we are now. So I thought I'd spend the rest of the time, assuming that I have some. Yes, I think I do. To talk about the normal brain vasculature, where the blood-brain barrier is intact, and where you actually can only hope to target with this method the endothelium or maybe pericytes, maybe even the neurovascular unit, but not beyond. Looking at the literature on peptides that people have reported to cross the blood-brain barrier, it struck me that they had this motif RXXR in a number of cases. 3 are listed here. This, of course, has not just that, but every 3 arginines would form this consensus structure as well. Now, we had identified this consensus structure earlier in some of our tumor screens. And what we noticed was that in some screens, particularly if we did them ex vivo using tumor cells, or even in vitro using tumor cells as targets, that we, somewhat annoyingly to us first, tended to find only these kinds of sequences in every peptide. And usually the second basic residue was C-terminal in them. And we actually then found that if we took one of these peptides and put it on quantum dots in this case, that the quantum dots were very effectively taken up by the cells, as you see here. But even if we just added an amide group to the C-terminal carboxyl, or put another amino acid there, the effect went away. So we named this the C-end rule and abbreviated as sender. So those brain-homing peptides I just showed you then have sender motifs in them that are effective in taking things into cells if they are if the motif is C-terminal. We've also found a peptide that has a tumor homing, or actually several by now, peptide that has a sender motif. But in this case, it also has the RGD sequence, which we identified 30 years ago already as a sequence that binds to integrins. And it turns out that the alpha V integrins are specifically expressed in tumor vasculature and in other activated vasculature. And this peptide homes to tumors or tumor vessels initially based on this RGD motif. But it then, as we showed in a couple of papers, gets proteolytically processed here and becomes an active sender motif peptide. And these peptides bind to neuropilin-1. And when a peptide binds to neuropilin-1, it activates an internalization and tissue transport process where the peptide and cargo attached to it, it just goes in and out of cells. We know a lot about the system now, but I won't have time to go into it. We hope to publish that soon. So one interesting thing, though, about it that Hongbo Pang has found in the lab is that the system, the activity of this system is regulated by nutrient availability. You take nutrients away and the system becomes hyperactive. So its function probably is to transport something to a tissue that is in trouble, like a hypoxic, poorly vascularized area of a tumor. But we can use it to transport drugs into tumors. And amazingly, while this cargo can be directly coupled to the peptide, it also works if you just give the peptide and the cargo together. We get about a 3-fold increase in drug delivery into tumors that way. So something like this then is going on in this system. You have the drug and the peptide circulating here. The peptide activates this endocytic internalization pathway, which we have now shown, actually not just inferred, but shown is also an exocytic pathway. So the stuff can go from one cell to another. Now, getting then back to the brain, how might we be able to isolate peptides that penetrate into the brain? Now, it may be that the sender motif system would work the same way it works in IRGD, that if we have something, a peptide or a motif that brings a peptide to the brain vasculature, that the cryptic or sender motif is activated, and the peptide and the truncated peptide and its cargo go into the brain. But we can also just use our system in a new way to probe for, to screen for peptides that actually penetrate into the normal brain. And here's how it goes. And we have this working in tumors. So we do the phage screen as usual. But instead of perfusing just with PBS, we use low pH, which inactivates the T7 phage. So any phage particles that are exposed out here will get inactivated, and then they won't come up when we rescue the phage. And then we also use detergent to eliminate the endothelial layer, which gives us then the basement membrane under them and the parenchymal tissue. And now we have eliminated vascular homing peptides and should only collect the ones that have penetrated into the brain tissue. These experiments are ongoing, and I don't know if they will work, but I certainly hope so, because I don't think the peptides that are available now are particularly effective in targeting the parenchymal tissue in the brain when the blood-brain barrier is intact. So what I've shown you is that in vivo phage display can be applied to brain vasculature and brain tissue in general, and that it reveals new specificities there, or what we think are new specificities. I told you about the peptide that targets brain injuries, penetrating and traumatic, and that we hope have some evidence for might also target concussions, which could be really useful. We have, I told you about 3 peptides that accumulate in the hippocampal lesions in Alzheimer mice from an IV injection. And that one of them detects the change in the neurovascular unit of the AD hippocampus. Now, what I haven't mentioned yet is that we know that the brain vasculature differs from the vasculature of other tissues, but might the different areas in the brain also have specific vascular zip codes? And that's something we want to study, and that all gets very close to the, maybe closer than the rest of what we have done so far, to the brain mapping. I just remind people that the vasculature is a very important part of the brain, and mapping that might also fit into the brain initiative. And we hope to be able to contribute to getting through the blood-brain barrier. Thanks very much.
Thank you. Thank you very much, Erkki. That's terrific. Let's have some questions. If we could have the microphone right here, perhaps, to start.
So I'm very interested in the brain vascular unit, and I wonder why you think that the labeling that you see in the hippocampus isn't in the rest of the astrocyte, that is in what you're calling the parenchyma. Right. Because there are differences. Elena Pasquale over in your place, right, has shown that there are different tyrosine kinase receptors on astrocytes in some layers of the hippocampus. So yes, where your labeling looks more like it's the astrocyte.
Yes.
Which upon injury upregulates and expresses more neuropilin, and which is another associated protein with tyrosine kinase receptors.
Yes. What I perhaps should have emphasized is that we simply haven't had time to identify the types of cells that our peptides bind to. I showed you some structures, but that's not the same as showing that the cell you think is an astrocyte is one, and the cell you think is a neuron is a neuron. That work is being going on, is being done right now, but I don't have results yet. It's we're actually doing it in collaboration with the facility at UCS here at UCSD. They have been busy, so we haven't gotten that far. So that question is open. We, of course, would like to get into neurons and maybe the other 2 peptides that don't seem to stay in the astrocyte-looking cell get into neurons. If not, we haven't exhausted the method. I should emphasize the fact that all these Alzheimer homing peptides seem to detect something that is only in the Alzheimer lesions. So whatever the target is for them, or targets are for them, would be of interest. And maybe they are tyrosine kinase receptors, as you suggest. We'll have to see. Roger.
Erkki, I have 3 questions, but I'll ask them in order. First, what is the sequence of any of these peptides, or is that a top secret?
Well, yeah, we haven't patented them, so
Okay.
if I tell you, I'd have to kill you.
Okay, okay. Okay. Second, I suspected as much. What is the difference between the Casper and DAG peptides? They both seem to be derived from the same sort of screen. I don't understand.
Right.
I missed what is the difference between them.
Right, they just have a completely different sequence, and they have different homing properties.
But
The Casper only seems to home to the neuronal cell tract in the dentate gyrus. DAG does that too, but it, in addition, detects the neurovascular unit. So I think they are going to end up having different receptors.
Okay.
And the, lost my train of thought, so go to your next question.
Well, and you raised the interesting possibility that sender might explain some of the literature peptides, such as RVG or whatever, angiopep.
RVG,
and that is a very striking recognition that those past peptides have RXXR, but they don't have it at the C-terminus.
Right.
So do you really think that they fit the sender motif
Well,
when it's not at the C-terminus?
Right. This is how I see it potentially happening. The RVG peptide is known to bind to the acetylcholine receptor or transporter. I'm blanking on it now. Anyway, it clearly homes to the brain vasculature based on something different than the RGD sequence. And then in analogy with IRGD, which I showed you, and we have several other peptides of that nature, it then could get proteolytically cleaved. The RXXR sequence is a favored cleavage site for furins.
Yeah.
And those are very common enzymes at the cell surface, processing many proteins from proprotein to the actual mature protein. So maybe, and we do know that brain vasculature has neuropilin-1. It may not have as much as tumors, but it does have, it's positive for that. So everything should be there.
Yeah, but as yet you don't have evidence that these other peptides are actually proteolyzed before they work.
No, no, I was staying with the spirit of this meeting, telling the audience what we hope to accomplish, like the previous speakers have done.
I'd like to continue the tradition of double-barrel questions, too.
Okay.
So the first is, do any of these peptides work as isolated peptides? Have you looked at that yet?
Do any of them work as?
Isolated peptides, just one copy, as opposed to on a nanoparticle?
Yeah, all the fluorescence results I showed were just the peptide with the FAM.
You mean titrating down to one copy per virus? Is that what you mean?
No, just the free soluble peptide. Because I talked so much about the nanoparticle bound peptides.
Okay, so those were...
Good, we got to clarify that.
So the second question about the phage display screen for BBB penetrating peptides. So I can think of 2 ways that a peptide might show up as positive in the screen, at least as I read it from your slide. And one is by doing what you want, namely non-invasively passing through the BBB and winding up on the other side. But the other is by somehow destroying the vasculature or the BBB in some way and leaking out. Is there some kind of cute negative selection way to distinguish them? I mean, they're both potentially interesting, but I'm just wondering what.
Yeah, I don't think it is likely because we're dealing here with an extremely small number of phage particles, so that they would have some sort of a function, some sort of an effect on the cell, I think is really remote. So the only thing that could destroy something is if we so.
Targeting tight junctions would be an example.
Yes, that would be sort of what I proposed for the sender. That's possible for the peptides that actually end up homing there. But I was showing you data with the whole library where the vast majority of the peptides are inert, and yet there was a big difference. So I think it is passive.
What size silver nanoparticles, and how did you address agglomeration?
How do I address what?
Agglomeration.
Oh, that they would clump.
Charge agglomeration.
Right. We use different sizes depending on what we want to do. If they are around 90 nanometers in diameter, they're very good at increasing the fluorescence emission of fluorophores coupled to the surface. If Gary makes them smaller, like 20 nanometers, they are less efficient in that, but they have other properties, like the smaller the nanoparticle, the less likely it is to be taken up by the liver and such. So those are the parameters we use. Now, they are coated with Phage, so that keeps them as single particles.
Anything smaller than 20?
Sorry?
Anything smaller than 20?
No, well, no, either size we always coat them with Phage. And then the peptide, if we have a peptide, is attached to the end of the Phage chain.
But just the two sizes, 20 and 90?
Well, yeah, Gary can make them at any size. I just took two examples from the extreme ends of the scale that he has used.
Here at the back.
Is there a particular reason to choose silver nanoparticles? Because the tangles that you showed in the picture are pathologically, they're positive for silver staining also. So I was just wondering if the silver was an obvious choice for that, or would we be worried about the silver interaction with the fibers?
Okay. We picked them because they were around. They may not be, not all the properties of those particles can be used in our this particular system. The main reason why we like them in our studies is that they can be etched even in vivo. Any silver particle that hasn't entered into a cell, if we expose the cells or the tissue to the etchant, is going to dissolve. And because of the fluorescence enhancement by the silver, the fluorescence goes away. And so that's been very useful in our analysis of the sender system, which is a cell internalization system. Now, our controls show that this isn't non-specific silver staining because when we just used a control peptide coupled to a silver particle, these structures showed no staining.
But in the control subject, they don't have any tangles, so
no,
I mean,
I showed you one structure in the AD sample that got our control peptide coated nanoparticles.
Oh, okay, thank you.
And it was negative.
Hello. You mentioned there is a time window where the blood-brain barrier is disrupted after the injury. Can you maybe comment how long the time window is and whether it's localized to the injury region or whether it's like in the whole brain homogeneously?
Right. It's, and this is well known in the literature, the blood-brain barrier is specifically disrupted in the injury area. And many people use just IgG as a marker of the leakage of blood into or plasma into the injury area. So it very nicely localizes only there. Now I've forgotten the beginning of your question. Oh yeah, yes. We see some homing, although it's attenuated still at 24 hours. So the window isn't that long. Luckily, the way I wrote the DARPA project was that we promised to be able to target the injury at 6 hours after the injury. And because I figured that that's when they would have the soldier in a field hospital or whatever and be able to treat him or her. So that's where that time period came from, but it seems to be optimal, and the window, as far as we can tell, is pretty short. We're doing additional screens that we do a couple of days after the injury, but I don't have to be successful there. It's not in the proposal.
Could you please comment on how well your nanoparticle will be diffusing through brain tissue after they pass blood-brain barrier?
I don't know. All I can tell you is that when we wait for half an hour, well, actually, we have not done nanoparticle homing yet with the brain tissue. Those were all just fluorescein-labeled single soluble peptides. So I don't know. Well, it remains to be seen. That is one of the goals of this project, that DARPA is particularly interested in siRNA targeting, and that would require nanoparticles. So we'll have to get to that.
The last question here in the front.
Thank you. What sort of map? What would a map look like of the region-specific changes in brain vasculature? Like, what kind of map would that look like? Would it be in 3 dimensions, and how could you interact with it? And what changes would you look for?
Right. So we would look for peptides that home only, say, to the hippocampus, to the cortex, and down to whatever structures might have, whatever the smallest structures would be that have a specific vascular zip code. It may be that they don't exist at all, that every vessel in the brain is the same, but I don't think that's going to be the case. So we then have a collection of peptides that are capable of selectively homing to different regions of the brain. So in the map, they'd show up in different colors, I suppose. And of course, once we figure out what the target molecule is, that would be, I think, very useful for all kinds of applications. For one thing, then one could target treatments not just to the brain, but to a specific subregion of the brain.
Let's thank Erkki again for